So, hello everybody, good afternoon. My name is Lars Baltzer. I am the representative of physical organic chemistry. Physical organic chemists are interested in synthesis. We need to make molecules. We make many different molecules. We make complicated syntheses, but also simple ones. But we are primarily interested in what these molecules do. So that means we have interest areas bordering on material science. Helena Grenberg, who we will see in a minute, or in a few minutes, is a representative of that branch. And we have an interest in the bio area. In order to illustrate the physical organic chemist approach to biology or drug development, I thought I would give you simply an example. So, liver cancer is a very terrible disease. It hits mostly men. By the time you discover it, it is most often too late for surgery or transplantations, which would give you a very good prognosis. So by the time you are diagnosed with liver cancer, you are left with drugs. So this is a drug that is currently on the market for late-stage treatment of liver cancer. It's one of the very few. Of course, it requires organic synthesis to put it together. I would say it takes a skilled organic chemist maybe 2 to 3 months to put that together. But the problem is that this molecule has very severe side effects, as illustrated in the panel here. And the reason is that a cancer drug is a very aggressive compound. It's a poison. You want to poison the tumor, but in the process you poison another part of the body, in this case the skin. So sorafenib is taken up in the skin. 44% of all the patients who are given sorafenib will be taken off the drug in about a month's time because they cannot function. Some of them are bleeding from hand and feet. You can see here the effects on fingers, you see the skin, and you see an illustration of how the skin leaves or simply is lost from the foot or from the hands. So this is actually what big pharmaceutical companies such as Bayer, this is the best they can do. There are some follow-up drugs in the pipeline. They are very similar in side effects. And why is this? It is because, to some extent, within these big companies with quarterly reports and so on, you're stuck in traditional ways of how to develop things. Nowadays, innovation quite often happens in university groups and in small biotech companies. In a university laboratory, in a university close to you, a variant or a derivative of sorafenib was developed by a group of scientists, chemists, physical organic chemists, who were not so impressed by traditional ways of making molecules. They applied a strategy that would not be attempted in a big pharmaceutical company. They simply attached this molecule through a spacer to a peptide. And this now comes from a physical organic way of thinking. If you want to avoid a small molecule that is hydrophobic and is spread throughout your body, if you want to keep it away from certain parts of the body, how do you do that? What are the molecular interactions? How do you deal with transport through membranes? This is all a matter of interactions of molecules with other biological systems. It is a matter of understanding how these compounds interact also with proteins. So it required organic synthesis. You need now to modify this molecule, adding on a spacer, a short molecule. This is not something that you simply do. You need to be an experienced synthetic person, and then you need to conjugate it to a peptide, and then you need to think a little bit, what kind of peptide would you like to use to attach it to? Even so, this small group with resources insignificant in comparison to what Bayer has managed to put together a drug that shows absolutely no presence in the skin, while still being active in the liver hepatocytes. And this compound is now at the end of the preclinical trials, perhaps entering clinical trials, let's say, in 6 to 12 months. Not only would we like to know how we make these molecules that may keep us alive a little bit longer, we may also would like to know where do they actually end up in the body. And for this, we use so-called imaging techniques. That is a further chemical way of modifying these molecules. You put a lamp on it. In this case, these are optical measurements. These are fluorophores. You may or may not see the contours of a mouse here. And this is an illustration of the fact that this molecule is taken up very strongly in the liver, where it is supposed to carry out its action. So again, the use of chemistry, but thinking in terms of physical organic chemistry. How do we put something where we want it? How do we avoid putting it where we do not want it? How can we see where it is? What are our hands? How do we know what we have done? So these are techniques answering some questions. But the most important question now is, now we know what molecules we can make. They look different from the ones that we use today. But who is going to make these molecules? And the answer is you. You're actually the next generation of chemists. You did not grow up in a traditional pharmaceutical environment. You are free to think. You are now have a beautiful life ahead of you when you can learn chemistry and you can think, I would like to do a little bit differently. So you are the guys who will make me live longer. And I thank you very much for that. So, thank you.